Amongst all the different viruses that have been put on the table as possible candidates for causing MS, probably the most likely candidate is Epstein-Barr virus. For example, the study that I talked about that Dr. Banwell did in Toronto, Sick Children's Hospital, where children who were 13 years or younger, which is uncommon for MS, but not unknown. And so the question she was asking was, what is the spectrum of viral infections, common viral infections in this group? Not at all meaning to show that Epstein-Barr virus is the reason why it happens. And 85+% of patients in that population was Epstein-Barr virus positive. Notably, there were 5 children that were not positive but had MS. The results can also be looked at it in a different way, that maybe the assays, the commercial assays that were used to detect Epstein-Barr reactivity in this population was not of a sensitivity, and maybe if she had used a whole different approach to infection, maybe even those 5 kids may have had reactivity.
Yeah,
we don't really know that.
If I may, I think that's her impression currently. That was the insensitivity of the assays. It's truly compelling evidence that there is a relationship between Epstein-Barr virus and multiple sclerosis in all populations. And if you look at serologic studies of adults with MS, as Ram said, virtually every single patient has a is seropositive to Epstein-Barr virus, whereas in the general population, on average, it's probably on the order of 90 to 92%. Well, you say, well, that's not very different, 99.9% versus 92%. But when you're looking at tens of thousands of people, the p-value for that is extraordinary. And in fact, Doug Gooden, many years ago, did a meta-analysis in which he found that the p-value was out to the 0.00006 or some ridiculous number like that. So clearly there's something about Epstein-Barr virus and its association with multiple sclerosis. And I think that the therapies that we employ may eventually move towards antiviral therapies as opposed to immune-based therapies.
The virus itself is so complex that to just say there are 2 subtypes is probably erroneous, and that's probably the reason why now, 2 years ago, the international virology community got together, and now you have 12 types of Epstein-Barr virus. So that's a tall order. It's a tall order because this is such a complex virus, and it's not like you can just immunize with the spike protein equivalent of the coronavirus and get immunity. But that said, there are many situations like that. Probably the best analogy that I can draw is smallpox and vaccinia. Those are huge, complex DNA viruses, but we eliminated smallpox with vaccinia. And I envision a situation like that if we cannot produce a suitable vaccine where a killed version of type 2 virus, if that's how we would want to go, would probably eliminate type 1 infectivity and therefore MS, if that's the way to go.
Well, as I mentioned in the talk, there's some circumstantial evidence to begin with. One is that the epicenter for coronavirus research is the Wuhan Institute of Virology. So they have more types of coronavirus on hand than any other laboratory in the world. Secondly, there was evidence of people becoming ill at the Wuhan Institute of Virology in advance of the pandemic. And there was some evidence that sequences of the virus had been taken off a website that were germane to the virus that we subsequently know as SARS-CoV-2. Now it turns out that there's a component of that virus called the furin cleavage site that is in large measure responsible for the ability to infect human cells, and that cannot be generated very easily. There's a suggestion that this was genetically engineered, and that's why humans were so readily infected. And more importantly, with both SARS and MERS, when you looked at the animals in the vicinity of these infections, many of the animals were infected. So if you looked at the animal markets where SARS arose, 80% of the animals had the virus. They were unable to find any animals in the marketplaces, the animal marketplaces in Wuhan that had the SARS-CoV-2. So it's hard to say with certainty, and there's a lot of debate, but I personally sort of favor that this probably was an engineered virus. And I will say that when the WHO was in China, they met with something less than a fully receptive group of Chinese investigators. They weren't allowed to do everything that they wanted to do. And when they looked at serum samples for evidence of SARS-CoV-2 that had been collected in advance of the pandemic, where they were expecting to see a couple of hundred individuals who had seropositivity with flu-like illness, when they looked at those samples, they found none, suggesting that this was something that came on quite suddenly. What's been demonstrated is that indeed there does appear to be an attenuation of the antibody response following the administration of ocrelizumab, that the longer you wait from the time of infusion to the vaccination, it appears that the antibody response is higher. But he also, and importantly, demonstrated that the T cell response appears to be as robust, if not more robust, in that population. And that, mind you, is a very important component of eliminating the virus from infected tissue.
The part that I enjoyed listening to and learning from the vaccination part of it is, the fact that the CD8 cytotoxic cells are robust, like Joe mentioned. That's a major viral clearance pathway, and so the fact that you in fact it was even higher than normal in the B cell depleted people. So when patients ask us, should I get vaccination? The simple answer is, irrespective of what you're on, yes. There was actually a paper that came out, from Israel about how we should not recommend the use of S1P1 patients getting the vaccine, and I think that's wrong. I don't think that's appropriate because, Amit hasn't studied S1P1, but I think you'll find some immune response to the vaccine even in that population. We have patients who have been on S1P1s that the first 2 shots didn't do anything in terms of antibody, and the third produced an antibody response. And that goes back to, again, the T cells having been primed and ready for, in other words, it took them a little longer to get to where we want them to be. So I think, even with anti-CD20, even though the antibody response is low, I think the recommendation is get the vaccine, get the booster, and get all the advantages that you can. And even though you may not really see an immune response by way of antibody, you are generating cytotoxic cells that clear the virus. So it's important.
I agree.
One of the things we need to realize is that not all S1P modulators are the same. They each work in a little bit of a different way. Sure, the general category or mechanism of action is relatively similar, but they do work on different receptors. I think the more that we continue to study these, the more safety data that we're going to have for our patients. But also the more efficacy data. Some of our most recent clinical trials have shown, yes, that the less receptors that are hit, the better we are in terms of efficacy or targeted efficacy. But I think the more that we continue to look and the more longer term data that we have on all of these therapies, the better off we're going to be to make informed decisions with our patients. The first S1P requires generally, for the most part, requires a first dose observation based on its mechanism of action and the number of receptors that it targets. And with the two most recent, again, unless there is a rationale or a comorbidity that a patient has, there really isn't a requirement for a first dose observation. So unless there's a requirement, or unless we're concerned, perhaps somebody with a uncontrolled asthma, somebody with longstanding respiratory issues that we might be concerned about, somebody with a pretty strong history of cardiac issues, whether they're stable or not, we may choose to do a first dose observation even if we're using one of the therapies that don't require it. And one of the things that we heard Dr. Bob Fox today talking about when he presented the Kurtzky Memorial Lecture to open our CMSC this year, he was talking about progressive MS, and he talked about disease-modifying therapies and different treatments and what's approved and what's available. And he talked about the role of the S1Ps. And I think one of the things that I took away from that presentation was that there are two different ways really to look at progression in MS, whether it's a degenerative or secondary to inflammatory in lesions. And he clearly said that there is a role for the S1Ps when we're looking at lesion-driven progression. Yes, I do. I've had experiences particularly with the first FDA-approved S1P modulator in the United States, when we were hard-pressed to have other options for our patients based on, for example, their JCV status and a high index and things of that nature. And so we took a deep breath and said, let's give this stuff a try. And we were amazed at how well it worked in our patient population who had a high amount of MRI activity, had a high clinical burden of disease. And so as a result of that, we have used it more and more often in those situations. And not just the first one, we have used all of the available S1Ps to date. And while I know there may be colleagues that would disagree with me in terms of whether these are high efficacy or not, I've seen it work in patients that I felt needed high efficacy therapy, so that has given me my own general confidence. I think if you look at the patient population and the breakdowns in each of the pivotal trials, you'll see that there were subgroups of people that had high efficacy disease and needs for high efficacy disease activity. And I also think that there are options that we might need to consider in terms of people's inability, for example, to get IV infusions, whether that's venous access, cost, transportation, COVID issues, whatever it is. And there might be other issues that would preclude them from taking other kinds of therapies. So here's a class of oral therapies that I say is worth a try in my highly active patients. And it's incumbent upon us as clinicians to appropriately educate our patients. One of the things that I find really important when I'm educating patients today in the era of COVID-19 is to first of all start off with how the immune system works, because many are very confused. They've thought all along they have an overactive or an underactive immune system, okay? And so they think they're immune suppressed. Well, that's not the case. We really need to talk about the fact that in MS, the immune system is overactive in various different ways. And what we try to do with the disease-modifying therapies is specifically target that overactivity. And each of these disease-modifying therapies works differently. So we start off helping them clarify what the immune system is and what it does. And that's important now because people are going to frame that in terms of COVID-19. Whether we bring it up or not, that's how they frame it. once they have a better handle on what the immune system does, we can talk about mechanisms of action.